I am a physiologist. It is the job of a physiologist to understand biological phenomena at all levels of organization. We focus on blood pressure, and we study it through multiple means, from whole population trends down to individual genes. The two biggest contributors to determining a person's blood pressure are cardiac output and resistance. We focus on blood volume and the control of blood volume, and a lot of that happens in the kidney. My most important job is passing on accumulated knowledge. During the day, your kidneys process about 25 moles of salt. So that's about the amount in about 2 Morton salt containers, so you have something to visualize. About 65%, 70% is absorbed in the proximal tubule through the sodium glucose cotransporter and the sodium hydrogen antiporter. About 25% in the thick ascending limb through the sodium potassium 2-chloride cotransporter and the sodium hydrogen antiporter. About 5% in the distal convoluted tubule through the sodium 2-chloride cotransporter. And the part that is mostly controlled by aldosterone, most directly controlled by aldosterone, is through the epithelial sodium channel in the collecting duct. One of our interests in the lab is the epithelial sodium channel, which creates some of the osmotic gradient, which drives the reabsorption of water and controls blood volume. We use Xenopus frog oocytes to express the protein. And then we can do techniques like single channel patch clamp, seen here, wherein we have a single channel in the interface between the glass and the oocyte. And we're able to record the actions of one molecule, one protein molecule opening or closing to allow the flow of current. In this example, there are multiple molecules, multiple protein molecules in the patch. Another technique we can do is 2-electrode voltage clamp, where 2 glass electrodes pierce the oocyte and allow us to measure the aggregate motion of ions across the membrane of the entire cell. These techniques allow us to look at what's happening at the level of the individual protein molecule or the individual cell. Other techniques allow us to look at tissues, organs, or even whole organisms. Molecular biology comprises a set of techniques which allow us to test things like protein size, relative expression, and processing. And really, molecular biology is integral to everything else we do in the lab. I hope you've learned a little physiology and what it's like to be a physiologist.